Hello dear learners. In the previous lecture, we discussed about the concept of RNA polymerase. What are the functional subunits of RNA polymerase? And what is the main role of RNA polymerase to carry out the process of transcription? So what is that? We will revise in just short time, that is, what are the different subunits of RNA polymerase and their function. RNA polymerase is an enzyme that is required to synthesize RNA in the direction of 5' to 3' by using DNA template strand that reads in the 3' to 5' direction. RNA polymerase that adds ribonucleotides like ATP, GTP, CTP and UTP, that is uracil, instead of thymine, or that is replaced by thymine. The prokaryotic RNA polymerase, it is a multimeric enzyme, that is it is made up of many of monomers, like it is required 6 subunit to form a particularly active RNA polymerase. Which are they? They are 2 identical alpha subunits, 2 non-identical beta and beta prime, and omega subunit, and the 6th one, which is very important factor, that is sigma factor. 2 alpha, beta, beta prime and omega. These are the core enzyme. When this core enzyme attached to the sigma subunit or sigma cofactor, it will lead to form holoenzyme, that is the completely active RNA polymerase, which required to carry out the process of transcription. So this is the revision of RNA polymerase or the structure of RNA polymerase. We know the function of different subunits, like alpha subunits. They are required to determine the DNA or the same sequence that need to be transcribed. Beta and beta prime, beta is required for catalyze the polymerization, that is addition of nucleotide, ribonucleotide. Beta prime is to unwind and to open up the DNA, like expose the DNA template strand for the process of transcription. Omega, the function is still not known. Sigma factor that required to recognize the promoter or the initiation site where we are going to start from the that process, that is transcription, or the promoter sequence is recognized by sigma factor. So now we are going to start about the transcription in prokaryotes. There are 3 important processes to carry out the transcription that is initiation, elongation and termination. इन इनिशिएशन फेज, इनिशिएट करना मतलब क्या, आपको वहाँ से प्रोसेस स्टार्ट करना है. सो आरएनए पॉलीमरेज जो है वो रिकॉग्नाइज करेगा कि यहाँ पे प्रमोटर प्रेजेंट है. अभी इस प्रमोटर पे वो जाके बाइंड करेगा और वहाँ से वो प्रोसेस स्टार्ट कर देगा ट्रांसक्रिप्शन की. फिर इन डिटेल्स इन दिस लेक्चर वी आर गोइंग टू डिस्कस अबाउट इनिशिएशन प्रोसेस. So initiation that involves the interaction of RNA polymerase with the DNA at specific site or the sequence of DNA. ये जो प्रोसेस होने वाला है इनिशिएशन, उस प्रोसेस के लिए RNA polymerase जो DNA है उसके कुछ स्पेसिफिक साइट पे जाके बाइंड करने वाला है, कुछ स्पेसिफिक सीक्वेंस पे जाके इंटरेक्ट होने के बाद वो बाइंड करेगा. The sequence of DNA needed for RNA polymerase to bind the template and accomplish the initiation reaction defines the promoter. जहाँ पे अपना RNA polymerase जाके बाइंड होने वाला है उस सीक्वेंस को कहते हैं प्रमोटर, and that promoter sequence which is present on DNA. मतलब वही सीक्वेंस अपने को बताएगा, इंडिकेट करेगा कि यहाँ से आप प्रोसेस प्रोसेस जो है ट्रांसक्रिप्शन का वो स्टार्ट करो. किसको बताएगा? आरएनए पॉलीमरेज को. प्रमोटर आर द कैरेक्टरिस्टिक सीक्वेंस ऑफ डीएनए दैट डायरेक्ट्स द आरएनए पॉलीमरेज टू इनिशिएट द ट्रांसक्रिप्शन. सेम, इट इज यूजुअली लोकेटेड ऑन द कोडिंग स्ट्रैंड. कोडिंग स्टैंड कहाँ पे था अपना, दिस इज योर कोडिंग स्टैंड, वहाँ पे ये अपना प्रमोटर सीक्वेंस, ये जो देख रहे हो आप, दिस इज योर कोडिंग स्टैंड, वहाँ पे आपका प्रमोटर सीक्वेंस वो प्रेजेंट रहेगा डीएनए पे, ठीक है। Simplest type of promoters that can be found in prokaryotes. Two general types of sequence elements are found. One sequence element is believed to promote initiate binding of RNA polymerase, and other usually has the high content of adenine and thymine. So this sequence, अब ये जो sequence है जहाँ से initiation होने वाला है, वो sequence present होता है कुछ -35 to -10 बेस पेयर जो प्रेजेंट है ट्रांसक्रिप्शन के लिए लगने वाला है डीएनए स्टैंड पे, वो वहाँ पे प्रेजेंट होता है. वी विल सी दिस इन द फिगर. देयर आर ऑन कोडिंग स्टैंड इंडिकेट्स डुप्लेक्स डीएनए रिक्वायर्ड फॉर ट्रांसक्रिप्शन. सो अभी यहाँ पे आप देख सकते हो कोडिंग स्टैंड है, वहाँ पे आपका प्रमोटर सीक्वेंस प्रेजेंट है. यहाँ पे आप देख पा रहे हो लेफ्ट साइड जहाँ पे एरो दिखाया गया वो आपकी जो साइट है वो है अपस्ट्रीम साइट. राइट साइड जहाँ पे एरो दिखाया गया है वो है आपकी डाउनस्ट्रीम साइट, ठीक है. तो जो ट्रांसक्रिप्शन यूनिट है, जो स्टार्ट पॉइंट है, वहाँ पे आप देख सकते हो +1, उसे बोलते हैं INR सीक्वेंस, इनिशिएटर सीक्वेंस. 3' to 5' ये हो गया आपका टेम्पलेट, जहाँ से आपका जो भी सीक्वेंस है वो कॉपी होने वाला है. 5' to 3' ये आपका जो है ये है कोडिंग स्ट्रैंड, जहाँ पे आपका प्रमोटर सीक्वेंस प्रेजेंट है. तो प्रमोटर सीक्वेंस जो होता है वो प्रेजेंट होता है -10 to -35, यहाँ पे आप देख पा रहे हो. और वो सीक्वेंस में कुछ 6 बेसेस जनरली आर प्रेजेंट, दैट इज -10 रीजन जो होता है उसे बोलते हैं टाटा बॉक्स या प्रिब्नो बॉक्स. क्यों उसे टाटा बॉक्स बोलते हैं? बिकॉज उसका जो सीक्वेंस है वो है TA, TA एंड AT. और जो अनदर सीक्वेंस जहाँ पे आप अपस्ट्रीम पे देख पा रहे हो -35 के रीजन पे, वहाँ का सीक्वेंस होता है TG, TT, GA, CA, ठीक है. तो ये सीक्वेंसेस प्रेजेंट होते हैं. टाटा बॉक्स ये लाइक प्रमोटर सीक्वेंस जैसा है, ओके. So in the next slide we'll see about the initiation process. So in the initiation process, the RNA polymerase that recognizes these promoter sequences, that is promoter sequence is in between -35 to -10. -10 sequence is also known as the Tata box or Prigno box. It is from the initiation sequence or initiating point. It is in the upstream of direction. You can see that this is in the upstream in the direction from the starting point. Starting point is also known as INR sequence or initiating sequence. From this, in the direction of upstream, there is a Tata box and the promoter sequence is present in between -35 to -10, where there is binding of RNA polymerase will take place. So you can see that here is the RNA polymerase. In this RNA polymerase, the sigma factor is attached and it is known as holoenzyme. In this holoenzyme, it will going to bind this promoter sequence in -10 to -35. This is the promoter sequence. When there is a no binding of the RNA polymerase to this complex is known as closed complex. So initiation process, it will be carried out in the 3 step: closed complex, open complex and the promoter clearance complex, ठीक है. So for this purpose, when there is no binding of RNA polymerase to the promoter sequence, so this is your closed complex. After that, when there is a binding of RNA polymerase to the promoter sequence, you know that sigma factor, this will going to recognize the promoter sequence, then it will facilitate the, or it will influence the binding of RNA polymerase to this sequence. After that, you know there is a beta prime subunit which is present in the core enzyme of RNA polymerase. that carry out the process of unwinding of DNA. So unwinding of DNA you can see here, and is also known as open complex. So after that, there is a beta subunit is also present in the core enzyme of RNA polymerase that will carry out the process of polymerization. That is, it will going to add the nucleotide. What type of nucleotide? That is ribonucleotides. So after that, there is a promoter clearance. When there is a release of sigma factor, the last step is promoter clearance. So this is about the process of initiation of transcription in prokaryotes. So if you have any doubt, you can ask me in the comment section. Till then, thank you.